My next talk is at 7:30. So I'm not going to talk very much about our science. That will be in the next talk. So if you want to see that, just join me when I dash out of here. We are interested in the nervous system, and we're trying to attack the issue of the complexity of the nervous system, try to understand how the nervous system develops and how it functions by using single-cell technologies. And this is just a beautiful drawing that's about 100 years old of what you can expect to see in the brain, which is neurons in black, various forms of glia and immune cells in green, and then the vessels in red and blue, which are, of course, composed of cells. We, several years ago now, came up with this idea to use a very straightforward strategy to analyze complex tissues. You take cells out at random. We don't like to select cells. We just take everything. You do single-cell RNA-seq on it, and then in the computer you cluster this data to discover cell types. And initially it wasn't really obvious that it was going to work, but it actually works beautifully. And we worked on all of these various steps here to make a nice workflow. Here's a paper we published last year where we applied this idea to the cortex and hippocampus. We isolated single cells, we ran them one by one on the Fluidigm C1 system, or actually then in batches of 96, sequenced them, and in the end we had about 3,000 cells that we could cluster and discover 47 molecularly defined types of cells in these tissues. So this is extremely powerful. This was a lot of work. It's probably 5-6 months of doing this thing over and over again, because you can only get about 45 or so cells per chip, 45 good cells per chip at the time on this instrument. Here again is what you would expect from such an experiment. You would find a lot of various forms of neurons, excitatory neurons, inhibitory neurons, vessels that are composed of endothelial cells and pericytes, and then the glia cells, astrocytes, microglia, oligodendrocytes, and the ependymal cells. This is what the data looks like. You have 3,000 cells by 5,000 genes, and this is a heat map showing gene expression. So this is the type of data that we work with all the time. And it's a very complex data set, but basically what you do is you go in here and check, what are these genes, and you figure out, using the genes and a lot of reading of literature, what are all the different clusters that you see here. And this is what we came up with in that paper, basically showing that we can rediscover all the major cell types. And the real power is that you can then dive in, and you see there's a lot of heterogeneity within the major neuron classes, and in those classes we were able to define all the major excitatory and inhibitory neurons. Anyway, so the way we go about it generally is that we, as I said, we look at, we just let the data tell us what it is by clustering it, and we obtain these clusters here. Here I have these 9 major clusters of cells in a t-SNE plot. And only then do we go in and look at markers that have some known function. So here are examples of markers that tell us what all these clusters are. For instance, Thy1 is a neuron marker, so it tells us that all of these cells are neurons. And GAD1 is the enzyme that makes GABA the inhibitory neurotransmitter. So therefore, these are the inhibitory neurons, and so on and so forth. That's just a little bit of background. We've been working with Srini and his team and applying the ICELL8 system to this challenge. This is our setup in our lab. This is the dispenser, this is the microscope. It's really on the same bench. I don't know why we cut out about 10 centimeters here in between, but it's really just next to each other. As Srini showed, it's a 5,000 well chip. And ideally, you could get 1,900 cells in the system. But of course, you have not only cells in your suspensions, you have debris and you have all kinds of junk. So you could expect maybe to get 1,000 cells if you work with primary cell suspensions. We're pretty happy with the instrument. It's easy to operate. The throughput is, of course, a great advantage. We like that you can do imaging and positive cell selection, meaning that you can actually decide which cells you want to work with. This is the same as we have been doing on the Fluid MC1 system, and for us it's really important because then we don't have to sequence all the junk. And sequencing is getting to be the major cost of single-cell analysis, so that's a really important feature. And it's convenient because, of course, the chips are pre-barcoded, so once you have done the RT step, you just pool everything and you have only one library to prepare. So the first experiments that we've done that I'm going to show you data from here today is the one cell line, U293, and then 2 Cortex samples. So these samples were prepared in exactly the same way as we did for the published paper. These are single-cell suspensions that are taken fresh out of mice and used immediately, so they're not frozen or anything. In these experiments, so these are 3 different chips, and we got this number of cells after first imaging and being quite selective, trying to get rid of debris and things like that, and doublets, of course. And they're also selected to have 1,000 molecules of mRNA per cell in the sequencing data. So these are the numbers that we actually work with then. For some reason, we ran each of these on 2 lanes on HiSeq. I think we probably underloaded them a bit because we got much fewer reads than you can get on a HiSeq. So these are the total read counts for each of these experiments, and these are the number of reads per cell. In the Cortex paper that we published, we had about 1 million reads per cell. And of course, you can work with much fewer reads per cell, depending on your question. But if you really want to have high resolution in subtypes of cells, I think it's still necessary to get towards 500,000 or 1 million. This is what our cell suspensions look like. I'm just showing you this to show you the challenge. It's not easy to know what here is a cell and what is debris. So we would, of course, select these beautiful cells. This might be an endothelial cell. They usually look like little bananas. This might be junk, or it might be a very small glia cell. We don't know. So this is just what you have to work with when you work with primary cells. In these experiments, we were quite selective. So I think we undersampled the glia cells a little bit. This shows, this is a plot of, on the vertical axis, the number of detected molecules, so UMI counts per cell, as a function of number of mapped reads. So you can see that we probably haven't saturated these libraries, so you can sequence more and get more molecules per cell. There's still a slope to it. And there's also quite a lot of variation. So there's actually a very large number of lines here, of course, because the total number of cells is very large. So these few up here are a bit of outliers. It's possible that these guys up here represent doublets or a piece of debris that we didn't select away. It's possible. Mapping these reads to the genome, it looks very good. We get almost all of the reads mapped to exons in the sense orientation, as you would expect. A few mapped to splice junctions in the sense orientation, again, as you would expect. Some reads map downstream of genes, defined as the 1 kilobase downstream of the annotated poly-A site. This is very normal. It happens all the time. It's because the gene annotations in the databases aren't very good. Not much maps to the upstream region. This is because we have actually re-annotated our upstream regions based on all of our Fluidigm data. And there's only a little that maps to introns. The 5' 3' distribution is more or less what you would expect. So this is the 3' protocol, as you probably know. So you would expect reads to bunch up towards the 3' end. So these graphs show for genes of different lengths the distribution from 5' here to 3' here. So all the genes are sort of stretched to this interval. This is just a percentage distance from the 5'. So very short genes, of course, since they're very short, the reads map sort of in the middle. And very long reads, they map towards the end. But actually, it's about the same distance from the 3' end, probably. The number of detected molecules is also pretty good. If you look at the cell line here, which happened to be the deepest sequenced, so at 500,000 reads per cell, we get from 10,000 to a bit more than 100,000 UMIs per cell. This is actually very good. I say based on my experience with the brain, so I have no idea how many molecules you're supposed to have in U293 cells, of course, but these cells are probably large and juicy. In the Cortex samples, we had a bit fewer reads, and let's take this one over here. We have 300,000 reads. See a sort of similar number of molecules per cell. It's shifted towards a bit lower numbers. But we would expect to have around 20,000 or 30,000 UMIs per cell, and this is more or less what we're getting. So this is very good. We like to look at the noise characteristics, and this is also a way of selecting genes that are informative in your data. And the way we do it is by plotting the mean expression across all cells versus the CV, the standard deviation divided by the mean, on a log-log scale. And if you have very good molecule counting data, and if you have no variation whatsoever except just the variation you get from putting molecules at random in wells, then you would get this linear relationship. This is the Poisson distribution. And if you have very noisy data, or if there are other problems, then you will have a lot of dots that are off this diagonal, up here or sometimes down here. So this is an example of a very good CV versus mean plot. It shows that there are almost no genes that are really highly variable. And this is from the cell line, so you don't expect to have a lot of heterogeneity there, right? There's a few genes here, but that's all. You can also look at a Tisney plot to look at the distribution of cells. And again, plotting the U293 cells, there is absolutely no structure in these cells. They're all exactly the same, which I think more or less makes sense. This is selecting, I think, 500 genes, the most variable 500 genes from the previous plot. On the other hand, if you plot the Cortex cells in the same way, then you get a lot of structure. So each of these plots have the same layout. These are Tisney plots of all the Cortex cells pooled together from the two chips. And up here, I'm showing actin expression. So actin is, of course, expressed everywhere. But here, I'm showing a marker of neurons, STATMIN2. And you see these are neurons, and maybe these are neurons as well. And in contrast, PLP1, which is an excellent oligodendrocyte marker, is shown down here. So clearly, we have a separation of neurons, and we have oligodendrocytes down here. And so on and so forth. So Claudin-5 are endothelial cells, Aquaporin-4 are astrocytes, and GAD1 are the interneurons. So at the sort of top-level analysis, this data set more or less recapitulates the analysis that we did by Fluidigm last year. Although we have to make much more detailed analysis to see if we can reconstruct all the more detailed cell types. That would require much more work. We did identify some sort of batch effect, so you probably noticed it in the previous slide that, for instance, let's go back. The oligodendrocytes here seem to separate into several islands. And if you look at these islands, you see that they come systematically from one or the other chip. And the same with the neurons. So you have the neurons from chip 1 here and the neurons from chip 2 here, or actually the other way around. And this seems to correspond also to the total mRNA counts. And I think this is, at least to a large extent, an effect of the very different sequencing depths. So one chip here was sequenced to 370,000 reads per cell, the other one to 100 and something reads per cell. And all these clustering methods are very sensitive to the total number of molecules that you detect. So you need to sequence to saturation in order to get rid of batch effects when you're using UMIs. One question that always comes up is the question of doublets. So I've tried to estimate it here. Of course, all these cells come from the same species, so we cannot do a real analysis of the technical doublet rate. But we can make the assumption that neurons and oligodendrocytes, there should be markers that are very clean markers of these cell types. And we know that there are some markers that are really clean markers. So STATMIN2 is a very clean neuronal marker, and PLP1 is a very clean oligodendrocyte marker. And this is a log-log plot of StatMin2 and PLP1. And you see you have a lot of cells that express StatMin2 and not PLP1, and vice versa. Here are the PLP1 cells that don't express StatMin2. And then there are some cells that seem to express both markers. If I put the lines like this, and remember that this is a log-log scale, so there's a huge difference between these cells here and these cells here. Right? You can actually count. This is 0, 1, 2, 3, and so on. 0, 1, 2, 3. So then I come up with a doublet rate of about 3.6%. And this is, of course, the sum of whatever is the technical doublet rate and our inability to perfectly separate neurons and oligodendrocytes. So in the brain, oligodendrocytes are actually wrapped around the axons of neurons, so it's not easy to separate them. And there will be some cells that are just, they probably look exactly like a single cell, but they're actually two cells. So my first impressions are that the data we get here looks on par with other methods that we worked on. We can rediscover the main cortical cell types that we're used to, and we can use the same markers and see the same things. The throughput is much, much higher than what we have been used to before. This was a single experiment, or actually 2 chips that more or less recreated what took us several months to do last year. And so next year, I guess we'll do the whole brain in 1 chip. Yeah. It's easy to prep a lot of cells, but you still need to sequence them. I think that's one of the take-home messages. So can't get around the fact that each cell needs to be sequenced to saturation to get good data. And I want to thank especially Srini and the Wafergen team, and Hanna in my lab, who did all the work, together with Amit and Peter, who did the bioinformatics. And then I'm done. Thanks.